I'm going to make a claim that vaccines are one of the greatest scientific inventions of all times. They've saved millions, probably billions of lives on our planet. So, how do these things work? How does going and getting a shot actually keep you protected against a virus? Well, in this podcast, we're going to talk about the answer to that question. We're going to talk about cellular immunology and how our body responds to foreign invaders. Let's get started. So I just want to remind you of the previous podcast when we talked about the different levels of immune response. Level 1, you may remember, are barriers, right? Our skin, our mucous membranes. That's our first level of protection against foreign invaders. The second would be the innate or non-specific. So these are general patrolling soldiers as well as macrophages moving throughout the body, white blood cells trying to engulf foreign invaders. This video, however, is really going to focus on level 3, adaptive immunity. And that really completes the triangle. That completes the 3 levels of defense. So we'll be talking about our lymphocytes, we'll be talking about antibodies, because these are our trained assassins. They're going to finish the job that the rest of the immune system started. Okay, so let's specifically talk about how this happens. So we have an invader in the body. How is that recognized? Because if you think about it, you've got cells in your own body that you don't want your immune system to destroy. So how does the body recognize invaders? Well, here's an invader. Let's say this is a virus. And that virus has invaded the body. How does the body recognize that that is an invader, that it's foreign? Well, it recognizes it because on the surface are these antigens. These antigens are like little cellular name tags that tell the body what's going on. So as we go throughout this video, you'll see antigens represented in several different ways. And I've sort of shown you 2 here, right? Here's what one might look like. And these are just artist renderings. It's really just sort of a glob of protein. So it really probably looks more something like that. Now, antigens are specific, and they will fit, as we will see, antibodies. So this thing right here kind of looks like a Y. That is an antibody. And antigens on a foreign invader fit into an antibody if your body has actually made those for you. So I wanted to start off with these words because they often get confused. Antigen is on the surface. It's a name tag. An antibody is something that your body creates to help fight off some of these harmful invaders. So, where do we get all of these cells in the immune system, right? How do we get all these white blood cells and B cells and T cells and antibodies? Where does all this stuff come from? Well, it all comes from stem cells, and we know that stem cells develop in the bone marrow. So if you go inside your bone and you look in the bone marrow, that's where a lot of stem cells develop. So there are a few different types, and it's not necessarily that you learn the long names of these. I'm just going to use this to show you. But one type of stem cell is called a hematopoietic or hematopoietic stem cell, depending on how you like to pronounce it. If you think heme, think sort of blood, iron, right? So these are the cells that are going to form blood cells. And there are 2 different types of hematopoietic blood cells, and they are myeloid and lymphoid. Let's focus on the myeloid ones first. Myeloid cells will all differentiate, okay? So once they look like this, they will further differentiate into these. Megakaryocytes, which actually will be used to form platelets. When we think platelets, think clotting. And then a lot of these other white blood cells, eosinophils, basophils, and neutrophils, those are all what we would probably consider to be white blood cells, leukocytes. They're going to go and they're going to be part of the immune system. Erythrocytes, you know as red blood cells. These are what carry your oxygen. We're going to focus in this video on these specific white blood cells here, monocytes. Monocytes differentiate into two types, dendritic and macrophages. We looked at this a little bit in the last video. So in this video, we're going to really focus on these, as well as the ones that come from the lymph. And those are our T cells and our B cells. Now, B cells also can differentiate into plasma cells, which create antibodies, which we will learn all about coming up. Okay? So in this video, we want to focus on these cells and these cells there. Okay, so I'm going to give you an overview of what I'm going to be talking about in this video. Now, this will probably seem like a lot, but I'm going to then break down every step that we go through here, and then we'll try to put it back together at the end. So we want to start with an immune response, all right? Well, we got our first layer of defense here. We got our skin. But let's just say a pathogen invades, and the antigen, right, what's on the surface of the pathogen, gets inside the body, okay? Well, the first thing that's going to happen is these macrophages are going to try to eat it, try to engulf it, right? The foreign body has come in, it's going to try to eat it. Then that will start two different cascades. We have some of this virus, let's just say it's a virus. This virus, some of it will come in, and those antigens will be floating in the blood. We call this a humoral response. When you think humor, think the fluids of the body. So this is in the blood and the lymph tissue. The other response that your body needs to initiate would be the response for the cells that are already infected, right? So if you've got a virus that comes in the body, it may make its way into some of your own cells. Well, so we then need to destroy some of your own cells because they're infected with the virus. So we've got two different paths here. One, to destroy the stuff in the blood. Two, to destroy the virus that's already in your infected cells. Now, this happens that the macrophage will alert the helper T cells. Helper T cells do what they sound like. They help. So the macrophage is known as an antigen-presenting cell. Antigen-presenting cell. Because it presents the antigen. It says, hey, I found something. Here's this antigen. You better get onto this. So it presents that to the helper T cell. Then the helper T cell activates both of these cascades. The B cell and the T cells are activated. The B cells, there are two types. The plasma B cells here. And what they're going to do is they're going to be involved in that very important process of creating antibodies that we talked about. Now, once we've gone through this process, right, this takes several days. We've created these plasma B cells, we've created these antibodies. It's probably good for the body that some of these B cells hang around and have a memory, so to speak, so that they're ready to fight off any infection in case you get it again. So we call those memory B cells. Now, on the other side, what about your body cells that are already infected? Well, then we need some T cells. The T cells, which mature in the thymus, by the way, this is why we call them T cells, and B cells mature in the bone marrow, if you remember that from the last video. These T cells, there are two types. Killer T cells, and these will go and directly attack the cells. And then, same thing, we have some memory T cells here that are going to remember. Okay, so this is a general overview. This might seem complicated, but what we're going to do is we're going to try to go through and we're going to try to break down all these steps to help your understanding. So let's start with the idea of lymphocytes. So lymphocytes basically are called lymphocytes because they're part of the lymphatic system. And we call those B cells and T cells. So let's look a little more deeply at B cells. B cells mature in the bone marrow. That's where we get the B. And these are involved in the humoral response, right? The fluids of the body. And the B cells will produce antibodies. So there's our bone marrow. That's where B cells are. B cells, if they could talk, they would say, I attack invaders outside the cells. So these are the invaders that are still circulating in their blood. They haven't quite made it into your cells yet. On the other hand, T cells mature in the thymus. That's where we get the T. And this is involved in your cellular response. They recognize antigen fragments or infected cells, and they're going to respond to them. So if a T cell could talk, it might say, I attack invaders inside the cells. So those are the ones that are already in your tissue, already in your cells, and they're causing you lots of problems. Okay, so B cells and T cells, B cells and T cells are actually just lymphocytes involved in specific reactions. All right, so let's really get deep into each of these. I'm going to start with B cells. We're going to go through that specifically, and then we'll end with T cells. All right, so B cells, I've said it several times. These are the ones in the fluid, in the blood, and the lymph. These are a specific response, and they create antibodies. And you remember antibodies fight antigens. So, what are the types? B cells, you remember, we talked about the plasma B cells, which create the antibodies. And this is a rapid response, short-term release from these plasma cells. Now, if we want a longer lasting response, like some long-lasting immunity, we need the memory B cells to be created as well. So these provide that long-term immunity for us. So once we get a certain disease or we get a vaccine against that disease, hopefully we got some memory cells that are going to stick around long-term to be able to fight off and give us a fast response if we need it. Okay? So the thing to remember about B cells is that they mature in the blood, they circulate in the blood, and they make antibodies. Really important things. So antibodies, let's get into them. Since B cells make antibodies, let's talk specifically about what antibodies are. So over here, you can see I got lots of B cells, right? There's a B cell, there's a B cell, and they've produced all these antibodies, right? So here's all these antibodies. Now, notice that they're different. Different B cells produce different antibodies because they are specific. So what is an antibody? An antibody is just a protein that's going to bind to an antigen on a pathogen or some freely floating ones, okay? And they're produced by the B cells. So let's say you have an infection of SARS-CoV-2, what causes COVID. So this pathogen has made its way into your body. Why are antibodies so important? Well, what antibodies will do is they will detect the antigen on the surface of the pathogen. So here's the whole virus, right? The whole thing is a virus. But on the surface are these antigens. So an antibody, you can see here, will specifically attach to the antigen. And they are unique and specific. Each of these B cells has roughly 100,000 antigen receptors. Now, if you look at it a little more detailed, remember I told you an antigen, we draw it like this, but it's really just a blob of protein, right? And the antibody, which is also a blob of protein, has an area here where the antigen binds to the antibody. We call it the epitope. So really, if you're looking up close, they're looking for a specific part of an antigen that they can bind to. So now you say, all right, I get it. Antibodies bind to antigens, or they bind to these viruses. So what does that do? Antibodies bind to it. How does that help the immune system? Well, that actually triggers a few things to occur because these antigens have bound to the antibody at this site. So this results in a B cell immune response. So let me walk you through this. This one's pretty complicated as well. So here we go. Here's our pathogen. It's got some antigens. And you can see here, same thing, SARS-CoV-2, there's a virus, there's its antigen on the surface. Now, what happens is, once this enters the body, it's tested by the B cells, right? Because I told you those B cells, they have like 100,000 different antibodies on their surface. So they're testing it. They're saying, okay, there's something foreign, let's test it. So you got all these B cells and their antibodies, and they're going around and they're kind of match up. Does it match? Does the key fit the lock? No, that one doesn't. Does this one? No, that one doesn't match. Does this one match? Oh, that one matched, right? So if one is recognized, if the specific antibody on the surface recognizes the antigen, that's the one that needs to be cloned, right? Because you had all these to begin with, but these up here are not going to really fight our infection. This is the one we want. That's the start. So after recognition, it's recognized. Oh, it fits the antigen. fit the antigen well, we need to go ahead and clone it. So we're going to make thousands of those, right? Lots of those B cells that match the pathogen. Okay, so now that those are cloned, they're going to make plasma B cells. So these will sort of differentiate into these plasma B cells. And if you remember, I said that plasma B cells are the ones that release the antibodies. So there they are, antibodies are released, and they're going to now be able to go and capture those invaders. So how is this working, Herder? How are these things? Yeah, I see that they're kind of bound to it, but what does that do? Well, I'm going to talk to you specifically in a moment about how the antibodies help provide immune support. So we've made these plasma cells. They've released their antibodies. We're capturing the invaders. There's one more important thing that has to happen, though. We've gone through a lot of work here. We've recognized, we've cloned, we've made some plasma B cells. We also need a few to be kept in memory or in reserves. So a few of these will stick around. They won't be differentiated into plasma cells. They'll stick around as memory cells in the reserves so that if this pathogen does come in again, it will be able to easily be attacked. Now, this takes about 10, 17 days for a full response. So for you to get full immunity, either from a vaccine or from having a virus, in this case, then you're going to need about 2 weeks to get this full response, to get all these antibodies and these memory cell reserves in place. So how do antibodies work? This is what I've been promising to say. There are a few different ways. I want to talk about 4. One is opsonization. Basically what happens in this case is that you can see the antibody here, right, this little Y thing, has attached to the surface of the bacteria or virus or whatever it is, and that has triggered phagocytosis, right? So it has triggered this macrophage to come in, eat the bacteria, take it in, and break it down. That's called opsonization. Another example of what antibodies can do is they can agglutinate it, meaning it can get all these, in this case it looks like bacteria, get all these together, agglutinate it together into one big wad. That way a macrophage can come along or some other immune cell can come along and take care of it. So it brings them all together, kind of gathers them all in one spot. Another thing that antibodies can do is they can actually activate the complement cascade. And you may remember in my previous video, we talked about this just being a series of proteins involved in the immune reaction. And then the last one is neutralization. So basically, if you think about it, these antibodies can bind to the antigens and essentially neutralize them so that the bacteria or toxin or virus, because the antigen is covered, it's blocked, the antigen cannot then be used to go and get into any cell. So it neutralizes them by blocking them from getting into any other cells. So now that we've talked about how antibodies work to protect us, let's now see about how these antibodies respond during exposure to a pathogen. So, for example, I've made a little chart here, a little graph, and over here we've got concentration of antibody, down at the bottom we've got time. So as you can see, after you have an initial exposure, so let's say you get exposed to a cold or a flu, well, it's going to take a while, several days, right, for that antibody concentration to build up. So this would be what we call your primary immune response, all right? So you're exposed once, your body mounted an attack, and there we are. Over time, those antibodies will slowly go back down. But if you get a second exposure to that same pathogen, look at the crazy number and the quick spread of those antibodies, how that concentration really moves up. So this, when we have a secondary exposure, we get a much stronger immune response in terms of antibody concentration. So these memory B cells that are holding on, right, they remember, they're allowing for this second response that's so quick. Well, this actually goes back to my opening slide, my opening information in this video about vaccinations. So how do vaccinations work? Well, really, what a vaccination does is it exposes you to a harmless version or some component of the pathogens, right? Either a weakened virus, or in the case of these modern vaccines, maybe just the messenger RNA or genetic material of that pathogen. What that does is it triggers some active immunity and will stimulate your body, your immune system, to produce these antibodies to fight off the invader. So even though you didn't get the invader, if I get a vaccine, my body's going to still build up these antibodies to fight the infection as if I had it. The good news is you don't have it when you get the vaccine. So you'll get a rapid response then if you get a secondary infection. So instead of being exposed, take that out. Your vaccine, in a way, is your initial exposure. So you're going to get an immune response. So then if you ever got a secondary exposure, you'd have the antibodies already made, ready to fight off, to latch on to that pathogen, and it'll be a quick response. So, are vaccines effective? Yes. Are they dangerous? No. Studies after study after study shows that unless you're having some allergic reaction or have a known problem to vaccinations, they are very safe for you. In fact, they will increase your livelihood, right? One of the big side effects to vaccines is a long life. Okay? So, just to kind of bring this in a little bit more, I wanted to show you a graph of what's happened before and after vaccine. So this is a graph, this top one, of smallpox in London over time. So smallpox killed a lot of people in London until we introduced the vaccine here. Edward Jenner develops that. And you can notice the gradual to almost nothing, right? Smallpox is nasty too, right? There's a small child infected with smallpox. So vaccines, thanks to science, thanks to vaccines, we have nearly eradicated that disease. Polio, another really horrible, debilitating disease. You can see some of the problems there. Jonas Salk developed this, and immediately, once we started getting these vaccinations, look what happened to this really horrible, horrible disease in the U.S. And measles is the same way. Now, science has given us this. It is our responsibility now as educated scientists to keep these low, right? There are people around the world who are deciding they're not going to do these, they're not going to take these vaccines anymore. And we're starting to see spikes in places around the world. So we need to be really careful to educate people and let them know about how vaccines work, what they do, and just how effective they've been at fighting disease. So that claim I made at the beginning, it was strong, but vaccines are one of the greatest scientific achievements of our generation. Okay, so vaccines made available by the antibodies. What if that attacker that comes in your body gets past the B cells, gets past the blood, and actually gets into some of your cells? Well, then we're going to need a different kind of response, right? These antibodies that have been circulating in your blood, well, we're going to need something a little different. We're going to need a trained assassin, and we call those T cells. So T cells are the cell-mediated response. These will get to your infected cells, let's say you've got a virus or a pathogen in your cell, or maybe you've got some of your own cells that are bad, right, like a cancerous cell. T cells will help to fight that. So I want to talk about three different types, right? You've got your helper T, your killer T, and your memory T. We've briefly talked about them, but let me go into a little more detail. The helper T cell does what it sounds like. When a dendritic cell or some macrophage ingests a virus, it will then go and alert the helper T cell. You can see here this antigen-presenting cell presenting to the helper T cell. And the helper T cell serves as a gateway, right? It helps other B cells and T cells proliferate and start to help fight the infection. So the helper T cell then, there's a few different types. The killer T cell, these are going to go to your body cells and attack those bad cells. But then just like the memory B cells, we need some memory T cells. These are ones that are going to hang around and they're going to remember and provide a much faster response. Okay, so when you think T cells, think these are the ones involved in my cells that are already infected. These are the ones that are going to fight those infections. And that killer T cell, like I mentioned, might actually go and fight a virus, a pathogen, or some of your own cells, like a cancerous cell, which is infecting your body. So how are T cells activated? So I mentioned this briefly, but there's a few different ways. One, let's say you got a virus, comes in the body. This dendritic cell has engulfed the virus, and notice that it's broken it down, but it's got some of these antigens, right? There's the antigen. So it needs to present that to a helper T cell. The other option is a macrophage, right? That takes in like a bacteria or something, breaks it down, and then it's going to present its information as well. The middle guy, right, to all of this is the helper T cell. This helper T cell is alerted from these antigen-presenting cells, which could be these or some others, and they start the rest, okay? So the helper T cell is an important middleman to deliver the news, to get these killer T cells and memory T cells activated so that they can go and fight this infection. So, how do T cells know that your cell's infected? Because you got a lot of cells in your body that are good. They're not infected. How does the T cells know that your cell is infected? Well, believe it or not, your cells in your body have got to sort of send up a little signal. Hey, I'm infected. I've been compromised, right? And they do this by moving the antigen to the cell surface. That alerts the helper T cell. So look here at my diagram. All right, so we got several good cells down here, right? This one's healthy. Yay! That's a healthy cell. This one not. Look at the viruses. Once these viruses enter, what happens is, breaks it down and it removes some of these antigens. And this little protein here, called the MHC protein, takes that antigen, moves it to the surface of the cell. It's sort of like taking it up to the top, up to the roof and saying, hey, look at what I found, right? So MHC, called major histocompatibility protein, there are several different classes, but one of the classes actually takes this antigen from the virus and it puts it out there and it says, look what I found. Well, then the helper T cell comes and says, oh, I see what's going on there, right? And binds to it, and then it will, of course, start its own cascade alerting the other T cells and B cells what's going on. So these MHC proteins are actually important because they're the things that take and put the antigens on the surface. So you might think of an MHC protein as sort of a snapshot of a cell. They're coming up to the surface and they're letting other cells know what's going on inside that particular cell in your body. Okay. So we've talked about how important helper T cells are. So now, naturally, I think I want to talk about what happens if something goes wrong with the helper T cells. Well, it's actually pretty bad news, right? HIV is a virus that infects certain cells. Specifically, it likes to attack helper T cells. Uh-oh. Well, helper T cells, we know, activate the rest of the immune system, the T cells and the B cells. So if I don't have helper T cells, I can't activate the rest of this. So HIV is called human immunodeficiency virus because it really makes your immune system deficient. So what's the difference here? The helper T cells are destroyed. It also can destroy some other T cells. So for example, here's a nice helper T cell, right? That's a T sub H. That's a helper T cell. Notice it's got this specific, what we call CD4 receptor on the surface. And what happens is the CD4 receptor, we know, is what HIV binds to and allows for entry into that helper T cell. So this virus is essentially attacking your helper T cells via the CD4 receptors. Now, what's the difference between HIV and AIDS? Well, AIDS is the syndrome caused by the virus HIV, right? And so when people die of AIDS, they really generally die of some other infection like pneumonia or cancer because their body couldn't fight off the infection because HIV did some really nasty things to those helper T cells. So again, HIV, nasty virus, and the reason it's so nasty is because it affects those helper T cells. So you've already seen this slide. I want to wrap this up by going through this one more time. If you think you've got a good grasp of this, no need to follow through, but I'm just going to summarize everything I said one more time, and I love this slide because to me it puts it all together. All right, so let's say that some nasty pathogen has invaded your body. Antigens inside. First thing that happens is the macrophage, also known as an antigen-presenting cell, will engulf that and start these two cascades going. You got something happening in your blood, and you've got something happening, there's the blood, got something happening in your infected cells. So what's going on? Over in the blood, you can see, you're going to get activated by helper T cells. So the macrophage will present its antigen to the helper T, we just talked about, and here it happens to be that CD4. It will then alert the B cells and the T cells. The B cells, there are two types we wanted to talk about. The plasma B cells, which make antibodies, and then you've also got the memory B cells. Over on the T cell side, you've got the cytotoxic killer T cells, as well as those memory T cells. Remember, the memory cells stick around to provide that secondary defense in case you are exposed again to a particular pathogen. Okay, so this video talked pretty in depth about the cellular immunological response to pathogens. Hopefully you learned quite a bit about your body's natural cellular immune response and all these special cells that are fighting on your behalf.